Hi, guys.
Hello.
Hey,
Good morning.
morning. Good to see y'all. Okay. So I wanted to talk to you a little bit about how things have been set up now. That's the syllabus. The timed online exams are part of your lecture grade, and those are always going to be due the following Wednesday, right? So we should be through 1 and 2 and 3 and 4. You should get to me by the end of today. Okay. There are Learn Smarts up there, which are homework. Those have all been designed as extra credit. Okay. The due date isn't until the last week, which is the 19th of May. Okay. But my recommendation is to do those prior to taking the exams. And that way you're going to be able to do better on the exams. Okay. So those are extra credit assignments. Your lab reports are obviously required. Okay. They're up there on Connect now, or not on Connect, on Compass, on the student portal now. You should be able to see all this stuff in the calendar. And then there's going to be a midterm and a final exam in both lab and lecture. The midterm and final and lecture is going to be through Connect, and the midterm and final in lab is going to be something that I will administer through the student portal. Okay, so that'll be something that you'll click on and take within the student portal, and it'll be available on the calendar as well. So I just want to kind of give you the layout of everything and how it's going to go, so that you know what and where to find all the material. And then as I give you guys these lab reports each week, I'm going to give you instructions about where to go to find the information that you need to complete them. Okay, so for the lab report from last week, you should have gotten an email indicating the website that you need to visit in order to do the Gram stain lab, which was what last
I didn't get that. I didn't get that email till I woke up this morning.
That's fine. That's fine. It's not due until 11:30 tonight. Okay, so you've got plenty of time, and it should be a pretty quick lab once you get the animation done. You just basically click on a link there, and it'll take you to the Gram stain tab in that little set of labs to perform, and then it'll tell you exactly what to do and how to do it, and you just walk through it. And basically all you're doing is raising your hand. Raising your hand. Sorry, Jablus, you raised your hand. What was your question?
Yes. I wanted to be sure, the Learn Smart, is that our lecture homework, or is that the one that's extra credit?
Well, it's extra credit and it's lecture homework.
Oh, okay.
It's both. Yeah.
Okay.
So
So the test is, I
again?
noticed that the test is given, okay, you know how you can take the test more than one time.
Right.
It's not giving you the higher grade. It's giving you an average grade, instead of, like, if I score an 86 on the second time I take it, but I scored a 78 the first time, it's not giving me my 86. It's giving you the like an in-between grade.
Yeah, I'll set it so that it gives you the higher one, but I'm able to see both grades and enter them by hand. So it's not a problem. You'll get the higher grade.
Okay.
Yeah.
Thank you.
Yeah, but the Learn Smarts, that's what they're for. They're to get you ready to take the actual exam. Yeah, Jablus, what's your question?
Okay, so we should be done chapters 1 through 4 and Learn Smart right now.
Right.
That's where
Yeah.
Okay.
Yeah. Yeah. And like I said, the Learn Smart due date isn't until the last week of class, but it's, if you do your Learn Smarts on time, you'll do better on the exams. Okay. So you kind of use those to train. Okay. Use those to train and get ready. In addition, I'm going to put up practice exams for you guys. All right. And practice midterm and a practice final that won't have any point value associated with them, but they'll help you get ready. Okay. Because the more you practice, the better you're going to do. And remember that you can always come in Friday from 5 to 7 with any questions that you got. Okay. So if you have some confusion about the lab or about the lecture, that's when you get with me and I help you out. Okay. And that's not just this class, that's for any science or math class you're taking. Okay. So just remember that when you come in on the Zoom session from 5 to 7 on Fridays, remember to identify your school and your class, because I have people from a lot of different schools coming in, and then I'll help you out with whatever you got. Okay. So today, it's week 3. We're going to talk about viruses, and we're going to talk about nutrition and growth in microbes. Okay. So we'll do 5 in here, get through most of that, and then we'll probably start 6 and finish 6 in lab. And then today's lab activity is going to be Kirby Bauer, which is on that same website as the Gram stain. And I will shoot you the report for that. And the instructions are on the website. So you should be able to navigate that fairly easily. And then I also need to shoot you the notes for 5 and 6, which I'm going to do right now. So let me take care of that before we begin. So I'm going to go here. I'll put them on chat and I'll also send them to you. So let's do this. Let's go to chat.
Is it possible that we can have the notes like maybe the day before, that we can have it
Oh
ready
yeah,
before we
yeah.
come
I'm
on?
planning to post everything today so that
Okay.
you'll be able to get to them on the portal. All right.
And I also have a question about the timed exams.
Yes.
I realized that the one we took this week, it had so many, labeling diagrams, scenarios, videos, stuff
Right.
like that. And I feel like the time was not enough.
Let's see, hold on here. File. Yeah, my computer. There we go. Hold on. Please. We want to go to here. Go to 5, 5 and 6. Page 5. Open. There we go. 6. Here. 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 There we go. Okay. So the notes are in chat. You can grab those out of chat if you're able to. Let me send you those now in an email. Okay. So let's do that. Here. Here. Da, da, that. This is 330, 331. 331, 21. There we go. All right. Take these out here. And do that. Attach. There's 5. There we go. And 6. There we go. Okay, 6. All right, so lecture notes just got sent. You should get those in a second if you didn't grab them in chat already. And for the time, what I'll do is I will go through and I will double the time that you have for each of those chapter tests, in addition to you getting 2 shots. So
Thank you.
help. Okay. So that. What's that, man? Do here. 5 and 6. Oh, Mary raised her hand. Mary, what was your question?
My question is, for the code, when we're gonna receive the code?
Oh, I'm gonna get that today from the rep.
All right, thank you.
Yeah, because this is week 2 of your 2 weeks. So yeah, I'm gonna take care of that. Yeah. Okay, so 5 and 6. Okay, and here we go for that. Okay, come on. Oh, there we go. All right, let's see if. Okay, let's go.
How can we see what our grade in the class is like right now to see like what our grade is?
I have to enter stuff first. I just got all the grade columns in last night, so I'm going to pull stuff from Connect and start loading it in today.
Wait a minute, I want to take one of my tests over before you do that.
But that's fine. That's fine. Anything you take again, anything you do twice, I'll go and grab that higher grade and get that assigned for you. So that's not a problem. Okay, so today I'm going to talk about something kind of, well, obviously we're very aware of it these days because it's all over the place, right? Which is viruses. Everybody's talking about COVID-19, but viruses in general they're an interesting kind of pathogen, okay? Because they may have been the first kind of disease-causing organism, not organism really, but let's call them particle, or let's just say pathogen, to exist on the planet. And the reason is that they're very simple in terms of their structure, right? All they are is genetic material inside of a protein box, right? And the idea behind a virus is that it's going to deliver its payload to a host cell in order to take it over and turn it into a virus factory. Okay, and there's different ways that the viruses can do that, right? Viruses can use either RNA or DNA as genetic material, and the RNA can be either single or double-stranded, as can the DNA be single or double-stranded. So in a virus, the rules are a little different than they are in a living pathogen, right? In a living pathogen, the genetic material is always double-stranded DNA, okay? But a virus does things sometimes a little bit differently, okay? And a virus is not technically alive because it cannot reproduce on its own. It has to have a host cell in order to make more copies of itself. So it flunks that criteria for being a living thing. Actually, the other components for life, other than maybe growth, are all satisfied by the virus, right? The virus can change, the virus doesn't really take in nutrients and doesn't really make waste products, and it doesn't reproduce, it doesn't grow in the sense of increasing in size, except when it's being built, but it can respond to its environment, and it can move, and it has a boundary, right? But a lot of the other criteria for life, the virus doesn't really satisfy, right? Where are viruses? Well, they're everywhere, right? They can attack any host, any living host. So they can go after animals, plants, fungi, algae, you name it. There's a virus in there. Seawater can have 100 million viruses per milliliter. You might think, boy, that's a lot. But viruses are very, very small, right? They're on the nanometer scale, right? 1 times 10 to the minus 9th meters is a teeny, teeny, tiny thing, okay? Much, much smaller even than a bacterial cell, okay? Which is much, much smaller than an animal cell. So you're talking really small. For a lot of years, viral infections cause was unknown. And this shouldn't be surprising, because even after the invention of the light microscope back in the late 1700s, you still couldn't see a virus, right? Because light waves are too large for something as tiny as a virus to affect the path of light. So you're not going to see that through a light microscope. So the viruses were still invisible to us, right? You know how people are. They only believe what they can see. So there's your problem, right? Pasteur postulated that a living thing smaller than bacteria could cause these diseases that were produced by something that was not visible to the naked eye. And he also proposed the term virus for naming these, and that's Latin, comes from the Latin for poison, right? So how did we come about this awareness of viruses? And it was very slowly, right? The electron microscope wasn't invented until the 30s, but we had evidence before then of something other than a bacteria or a protozoan causing disease. These two gentlemen, Ivanovsky and his colleagues, showed that disease in tobacco was caused by something other than a bacteria, a protozoan, or a fungus. Loeffler and Frosch discovered an animal virus that causes foot and mouth disease. Early researchers found that when fluids from host organisms were passed through a porcelain filter designed to trap bacteria, the filtrate, the stuff that came through the filter, could still cause disease. So we were able to filter all the known pathogens out of these liquids, but the liquid still had pathogen in it. So this was obviously smaller than something that we were aware of at that time. And what this proved was that a fluid that was free of cellular components still had stuff in it that can cause infection, okay? So the question becomes, what's up? What is this teeny tiny thing that makes you sick? Questions about viruses. Are they organisms? That is, are they alive? And the answer is they're in that gray area between living and non-living, okay? Did viruses play a role in evolution? Probably so, because one of the things we're going to find out that certain viruses can do is they can stick their genetic material into host genetic material, and then they can turn around and pull that genetic material out. And sometimes when they do that, they take a little bit of host genetic material with them, right? So they tear the chromosome up a little bit, and then that new piece of genetic material can hop into a virus, and then that virus can end up infecting another host cell. And then that genetic material from host number 1 can end up in host number 2, which means that we can move stuff around. It's like a subway for moving material from one host to another host, okay? And so that could even be from one species to another, right? So Did they play a role in evolution? Quite possibly, quite possibly, okay? What are their characteristics? How can something so tiny cause disease? Well, we're going to find that out. And is there a connection between viruses and cancer? And the answer is yeah. Okay, there are some definite known connections. Human papillomavirus, for instance, is known to cause cervical cancer, right? And the way that it does that is by introducing genetic material into normal genetic material and causing a change in the DNA that turns a normal cell into a cancer cell. So you call it a virally induced mutation. So the viral debate, right? Since viruses are unable to reproduce by themselves from the host, they're not living things. Oh, excuse me. And should be called infectious molecules. And the other side of the debate, even though viruses don't exhibit most of life processes, they can direct them and are certainly more than a lifeless molecule. Okay. So we describe viruses as either active or inactive, as opposed to alive or dead. So by active, we mean can cause an infection, right? And by inactive, we mean it can't. Okay. So viruses can infect cells and change their DNA. They can introduce new DNA into host DNA, and that changes it, and that's a mutation. This can change the way cells, tissues, bacteria, and plants have evolved, right? And we already talked about the fact that there's three possible outcomes from a mutation, right? You can have a good or a bad or a neutral outcome, right? A good outcome from a mutation is to provide that organism with some sort of tool that gives it an advantage, makes it more likely to live long enough to breed in a particular environment, right? A bad change makes it less likely that our organism will live long enough to breed, and a neutral change won't have any effect, okay? So it's entirely possible, in fact, probably likely, that viruses drove evolution, okay? Simply because they introduced differences in the populations that they infected. 10% of our DNA comes from viral DNA. So you're actually born, believe it or not, with virus instructions already inside you. And given the right set of conditions, those viruses can jump up and infect you from the inside out. It's kind of a disturbing thought, but there it is, right? That's some evidence that viruses have definitely driven evolution on the planet over time, right? Because they're in there already as evidence of them having gotten into host cells in the past, okay? So in some cases, right, when you end up with what you think of as a viral infection, it may not have come from the outside. It may have come from the inside, okay? So these guys are, you think of them like sleeper cells, right? They're already in there waiting to jump out given the right set of circumstances. 20% of bacterial DNA comes from something called phage, okay? Phage is the viral equivalent of what attacks bacterial cells, right? So even bacteria are victims of these unusual pathogens. And a phage, it does the same routine as a virus does, right? A phage is just genetic material inside a protein box that is going to land on a bacterial cell, going to introduce this genetic material and take over that bacterial cell, okay? In some cases, the bacterial cell will eventually explode from making all these virus particles. In some cases, the bacterial cell will produce the virus particles piece by piece by piece, slowly, as it drifts around inside the bacteria population. And then those viruses will go out and infect other bacterial cells, okay? We would call viruses obligate intracellular parasites, meaning what? They must have a host to reproduce, must have a host to reproduce. That, let's write that down. That's pretty important. Must have a host to reproduce. So okay. Must have a host to reproduce. Okay. They can't reproduce without the host because the host has all the other goodies necessary to put new viruses together, right? It can make new protein. It allows those proteins a way to interlock and assemble so that you make the box that you're going to put the genetic material for the virus in, okay? So the host is absolutely critical to make this happen. Because the virus relies on a host to reproduce, the virus genetic material is usually pretty short. Okay, it only needs a few sets of instructions to convert a host cell into a virus factory. So when you're looking at the length of virus genetic material, it's way tinier than even a bacterial chromosome. Okay. So very, very short set of instructions. Okay. So those are some of the properties of viruses, right? I won't go through every one, but basically what you have, right, at its core is a box of some kind. Okay, the box can have different shapes. We'll find out about those in a minute. And this is made out of protein. Okay. And in some cases, it may be cloaked in some host membrane, right? It's kind of a dirty trick that the virus can pull. The virus can steal a little bit of membrane from the host cell and disguise itself so that the immune system can't pick it up, right? Membrane. I mean, not in every case, but in some cases, it'll do this. Stop. Stop, 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 stop. Stop it. There. Membrane. Okay. And then inside, you've got genetic material. Same color. Yeah. Genetic material. Meaning what? Meaning it could be DNA or RNA. Which can either be double-stranded or single-stranded. Okay? That's basically it. All right. This is in some viruses. Okay. This here is in all viruses. Right. The genetic material is in all viruses. Okay. So there it is, right? Very compact pathogen. Okay. For a lot of years, animal viruses were classed on the host and the disease that they caused. But now we have new classifications that talk about the host and the disease and their structure and their chemical composition, how similar they are to each other by comparing their genetic material. The Committee on the Taxonomy of Viruses recognizes 3 orders, 75 families, and 283 genuses. Its plural is genera. Okay. So I'm going to talk about how viruses are set up, okay, how they're built, and what the different features are and what the features do. Okay. So structure of a virus, right? Size. they're some of the smallest infectious agents, okay? Parvovirus is 20 nanometers in diameter. Remember that a nano equals 10 to the minus ninth. This is really, really, really small. You can't see nanometer objects even with a light microscope, right? Largest viruses are about 10 times that, okay? Some viruses can be long, 800 nanometers, that's almost a micrometer, okay? But are so narrow that, again, you can't see them unless you have an electron microscope. Viruses don't look like cells, and they don't have any ability to make protein. And they're built from little pieces of protein that fit together like puzzle pieces, okay? So they have these unusual shapes, right? They look a little bit like Christmas ornaments, okay? The structure contains only the parts needed to get in the host cell and control it. So there's a coat, there's a core protein with genetic material. There might be an enzyme or two in there, okay? But that's not in every virus, that's just in a few. The capsid is the protein box that's around the genetic material. The nucleocapsid is the protein box and the genetic material. Naked viruses only have nucleocapsid and genetic material, while enveloped viruses steal a little bit of host membrane when they come out to disguise them. Spikes can be found on both naked and enveloped viruses. They stick out from the protein box that the genetic material is in, and they're hookups, okay? So they are how the virus locks onto the host and gets in. A virion is a fully formed virus that can make a new infection in a host cell. So he's ready to go. He's ready to latch on and take over. The capsid is made up of protein subunits that are called capsomeres. You can think of those as the puzzle pieces that will assemble themselves into a finished capsid. So this is basically a big protein complex that has all the levels of protein structure: primary, secondary, tertiary, and of course quaternary, right? Because these are polypeptide chains that are fitting together to make this big house. They're going to stick the genetic material in. Different types include helical capsids, icosahedral. So helical, they look like almost like tubes, right? And icosahedral look like Christmas ornaments almost, you know, Christmas ornaments with that in some cases are round, or some cases they have sides to them like a cube, or we'll see some examples. Enveloped viruses steal some host membrane when they come out of the cell to disguise it. And they can bub from the cell membrane, or from the nucleus, or from the endoplasmic reticulum, okay? And so there they are. I mean, these don't look anything like cells. They look really weird, right? You can see there's an example up here of a naked virus, right? It's just this tube, and then there's genetic material inside of it, right? That's a naked. Okay. There's an enveloped virus, right? So you've got protein fitting together here, right? You can see the spikes, right? And then you've got membrane stolen from the host around the outside, right? So enveloped. And then you've got another kind of naked. This is icosahedral. This is the one that looks kind of like a Christmas ornament, right? So see the spikes, right? See the shape, right? These are the hookups, right? These are the hookups. Spikes, equal hookups. And then over here, we got some envelope viruses, right? These are actual micrographs. All of these are micrographs, right? And so you can see what they look like here, right? There's a DNA core, there's the capsid, there's envelope out there and spikes. Okay? And this is a phage, the bacteriophage, right? Looks like a lunar landing module, okay? You got tail fibers, you've got tail pins and a shaft. There's the icosahedral head, there's the genetic material. And when this thing lands, it lands on the bacterial cell. And then this little thing just injects the genetic material, just like a needle, into your arm, right? It injects it into the bacterial cell and it just squirts out inside the bacterial cell. Let's be the host down here and takes it over, okay? And then the protein shell without its genetic material actually stays latched on the outside for a bit, okay, before it eventually gets shaken off when the bacterial cell explodes or as the bacteria kind of moves through space. Okay? So look nothing like a cell, okay? So it's important, right? No cellular structure. Right? This capsid, genetic material, and then sometimes membrane. Okay. So the genetic material in a virus can be either DNA or RNA, but not both. Okay. And the DNA or the RNA can either be single-stranded or double-stranded, but not both. Viral genome is tiny because you don't need a lot of instructions, right? Some examples, right? 4 genes in a hepatitis B virus, a few hundred in a herpes virus, right? They only have the genes needed to invade the host and to redirect its activity, right? So basically turn it into a virus factory. So I'm here, I've latched onto you, I shove my stuff into you, and my information now becomes part of your information and it turns you into something else, okay? DNA viruses can either be single or double-stranded, and the DNA can either be linear or circular, okay? RNA viruses can be single or double-stranded, okay? Positive sense RNA is ready for translation. Ready for translation, right? While negative sense RNA has to be copied into a complementary strand before translation can happen. Sometimes RNA viruses are segmented, where you'll have a few chunks of genetic material that are in there, and sometimes they aren't. Okay. Retroviruses carry their own enzymes to make DNA out of RNA. All right, now that's a special case, okay? And these are called retroviruses, okay? An example would be the AIDS virus. Okay. So the enzyme is called reverse transcriptase. And it gets that name because it does the opposite of transcription. It makes DNA from RNA instead of making RNA using DNA. Okay. And what's nice about that is that provides a drug target. So this is one of the few examples where we have a protein that's only in a virus, it's never in a healthy, normal cell, that we can attack. And if we attack it and only it, we can take down the virus without really hurting the host very much. So that's lucky. Okay. So if your problem disease is caused by a retrovirus, you have luck in that regard. So other stuff in the virus particle besides genetic material are enzymes, polymerases that make DNA and RNA, replicases that can copy RNA, reverse transcriptases that make DNA from RNA. But there are no proteins that do metabolism, okay, that make ATP from nutrients or that take the ATP and use it to assemble stuff, right? The host is doing all that work. The host provides the energy and the host provides the building blocks. Some viruses carry away stuff from their host when they emerge, such as transfer RNA molecules in some retroviruses and membrane in some viruses, right? The membrane is used as a disguise so that the immune system will attack it. Hold on just a second. I need to take this. Okay, we're back. All right. So we're going to talk about the life cycle of a virus, which is kind of a bad term to use. Just talk about how viruses make more copies of themselves, right? And then we'll talk about the different kinds of infections, and then we'll talk about lytic and lysogenic bacteriophage. And we'll get answers about what those terms mean. Okay. So viruses are tiny and they're able to capture the host's processes, metabolic processes, and change them into virus production. All right. So all the host is doing is making new virus. All right. Sometimes the takeover is so extreme that the host will eventually explode full of virus and just release it, and then that host cell will be destroyed. Sometimes the host cell will be allowed to survive and keep producing virus at a low rate. All right. And it'll still be able to function as a cell, but not a cell really that is normal, right? It's a virus factory cell. It's not a cell doing a job for the organism anymore. Okay. So the viral replication cycle talks about how the virus gets from host to host, right? What carries it around, then what it does to the host, then how it deals with the host's immune system. And then we're going to talk about how we control this. Okay. So these are the basic steps, right? Adsorption is what? Let's hook up. Right? Penetration, that's self-explanatory, right? That's getting in. Stop jumping around. Stop it. Stop it. Getting in. Right. Why are you doing that? Getting in. I'm gonna have to cut the camera so that the panel behave. Hold on. Getting in. Okay. Uncoding is basically releasing genetic material. Releasing genetic material. Sorry. Synthesis is making new virus parts. Okay. Assembly is putting the parts together. Release is letting the new virus out. Okay. The length of the cycle can be either 8 hours in some cases or 36. All depends on the type of virus. Right. So absorption, right? That's where we hook up. Okay. A virus is going to be, it's going to define its host range, meaning what it can infect. What it can infect. By what it can attach to, right? So these little spikes here are the hookups, right? And the hookups are only going to latch on to a particular membrane protein in a particular host cell, okay? So that tells us where it can go, right? Those are the keys that unlock the host, all right? Hepatitis V, for instance, goes after liver cells. Poliovirus goes after nerve cells. Rabies can attack different cells in mammals, okay, but not every single one. Cells that don't have the appropriate membrane protein aren't going to be hit by the virus, okay? So that right there is a place where we can do a drug target, right? We can attack these little spikes, okay? And if we can interfere with what they do, we can keep the virus from causing trouble. Okay? Tropisms are the specificities of viruses for particular tissues, meaning what? The virus will go after connective tissue, muscle tissue, nervous tissue, or epithelial tissue. Okay. So what kind of tissue do you tend to hit? Okay, and what sort of host do you hit it in? Penetration, that's getting in, right? The cell membrane of the host gets penetrated by the whole virus or its genetic material. Penetration can be through endocytosis, where the cell just basically grabs you and eats you when you come inside, and then you're going to be inside a membrane enclosed vacuole. Direct penetration is where you have fusion of the viral envelope with the cell membrane and then uncoating the enzymes in the vacuole dissolve the envelope and the capsid, and the virus then fuses with the wall of the vesicle. The genetic material is then in the cell cytoplasm and ready to be used. Okay. DNA viruses come into the host cell nucleus and are reproduced and assembled there. RNA viruses do all their work in the cytoplasm. Okay. But retroviruses turn their RNA into DNA, and their work is done in the nucleus and in the cytoplasm. Okay. So let's look at a double-stranded DNA virus, see how it does its work. Viral DNA comes into the nucleus where genes are transcribed into messenger RNA, because that's where those enzymes that do that work are going to be. The transcript moves into the cytoplasm, gets translated into viral protein that is going to be required to reproduce viral DNA. The host cell's DNA polymerase can be used here. In the late phase, part of the viral genetic material is translated into protein needed to build the capsid and other stuff. New viral genomes and capsids are put together, and the virus gets released in a process known as budding. Okay, so here you can see, right? Different viruses doing different things, right? Here's double-stranded DNA, right? Double-stranded DNA gets in, right? The double-stranded DNA is used to make messenger RNA in transcription, okay? So this is transcription. Okay, and then translation makes the protein. Okay. In a positive DNA virus, single-stranded DNA, we copy it to double-stranded DNA using a DNA polymerase. Okay. And then from the double-stranded DNA, we transcribe RNA. Okay, and then we translate that into protein. And then from the double-stranded DNA through the action of viral enzymes, we make more DNA to load into the capsid. Okay, so the capsid's being made out here in the cytoplasm. The new DNA is being made in the nucleus, right? And then we're going to learn about the other kinds, right? Positive RNA virus comes in, can immediately be translated and assembled, right? So this is translation. Okay. There's your protein, there's your capsid, nucleocapsid. Okay. And then the positive RNA through, again, viral enzymes is going to make negative DNA, negative sense DNA, and that's going to be used to make more positive sense DNA, right? RNA, I mean, and then that's used to do even more translation. So this keeps up the translation. In addition, this is going to be put inside the nucleocapsid, right? So here, this is translation, right? Translation also here, right? And then we assemble the capsid and load in the genetic material. In negative sense RNA, viral enzymes. Make positive sense DNA from negative sense DNA, and can make negative sense from positive sense DNA, right? And they can also make positive sense DNA that's used in translation. Translation. To make protein, and then we assemble the nucleocapsid. Okay. With double-stranded RNA, we've got both strands. The positive strand is going to be used to make protein through translation. And is also going to be used to make double-stranded RNA using viral enzymes. Okay. And then they get assembled into a nucleocapsid. And then finally, positive sense RNA can be reverse transcribed into DNA in retroviruses. This is reverse transcriptase. Okay. And then the DNA gets copied into double-stranded DNA by the host enzymes in the nucleus. And then that gets used to make normal messenger RNA, right? This is transcription.
Mr. Converse, for some reason, your share screen, I can't see anything.
Oh, hold on. There's let's see. Let me do this. Let's go desktop and share. And then let's go new share and do that. Can you see it now? Is it up now?
No, I just see the picture of you.
Can you see the virus picture with all the drawing on it?
No, I'm not seeing that.
Hold on. Let's go to desktop.
I don't know why, because I can see it.
You can? Okay, maybe it's
Yeah.
something I did. Give me 1 second.
Okay, can you see it now? Okay.
Okay, yes, I can see it.
Okay. All right. And then this is translation. Okay. And then we put the whole thing together, and that's the nucleocapsid. Okay. So all of that is dependent on every step where you see translation and transcription, that's all host doing that work, right? The other stuff where you're making DNA from RNA, or you're making RNA from RNA, those are virus enzymes that are doing that. But those virus enzymes are actually translated by the host, okay, in many cases. Sometimes the virus will come in with those, but oftentimes the host just whips that stuff out because there's RNA there ready to go. So you can see how if there's no host, right? No host equals no virus. Because the host is helping with so many steps here, okay? Totally dependent. There's all sorts of things going on in a lot of these that are not normal functions of an active cell, right? Like making RNA from RNA or making DNA from RNA, okay? Those are all things that you don't normally see. And that's one of the reasons why our immune system has something called interferon. human immune system. Okay, and interferon attacks double-stranded RNA, okay, wherever it finds it, and wipes it out, right? And that cuts down on the possibility of viral infection in other cells, okay? But it turns out interferon has to be made in tiny amounts because if we make too much interferon, it damages our liver, okay? It's hepatotoxic, right? So the body is clever about how it uses this very potent weapon, okay? So those are all the different tricks, right? The different ways that viruses can do their thing. Different genetic material, right, in some cases, and processes that only happen when there's a viral infection in many cases, right? So one of the things you noticed about this is that since the host does so much work, right, a lot of times when we design an antiviral drug, okay, can attack our own tissue. And why is that? Because some antiviral drugs are going to go after viruses that only use host processes or certain host processes, and that can eventually hurt us. Okay, so it's hard to make an antiviral compared to an antimicrobial, right? Something that goes after like a bacteria. And the reason is that the virus is using us against us, okay? Whereas in a bacteria, there are enough molecules and enough organelles that are only in bacteria that we can make a drug that only goes after them and leaves the rest of our tissues alone, okay? So this is one of the reasons why people complain, well, why don't we have drugs against viruses? This is why, okay? Because the virus is being tricky, okay? He's using our own tissue to defeat ourselves, right? And another thing that a virus can do that's really nasty is that some of these viruses can integrate and go to sleep, meaning that they can take their genetic material, stick it inside our genetic material and leave it there for an indefinite period of time, a long time. And it won't pop back out until the right conditions happen, okay? Sometimes the genetic material can be separate from our chromosomes, and sometimes the genetic material can be literally part of our chromosomes in some viruses, okay? And it'll sleep there, and that's a condition that's called lysogeny, okay? Okay, think of that as sleeping virus. Okay? And it's only when the right conditions happen that the virus jumps out and becomes what we call lytic, meaning productive or active. Any of you who've ever had chicken pox, right? There's a virus called herpes simplex, which is going to attack nerve cells. And it's going to make virus inside those nerve cells that erupts out of the endings of the nerve cells and onto the surface of your skin. And that's what makes those itchy blisters, okay? And then eventually what'll happen is that the virus will go to sleep and will live inside our nerve cells for the rest of our lives. And if the right conditions show up again, then that information can wake up and can cause a condition known as shingles in older adults. Okay, that's one of the reasons they recommend a shingles vaccine for older people, is to remind our immune system that we have this sleeping virus inside us, so that if it tries to pop out again, the immune system can deal with it before it actually produces the disease. Okay. And that turns out to be the way that we deal with most viral diseases is not by an antiviral, okay, because of the reason down here, but instead by vaccination. Okay. And the idea behind vaccination is to warn your immune system about viruses that are out there by showing it a piece of the virus, not the whole thing, and letting our immune system make tools to attack that piece of the virus. And then if the real virus shows up, because it'll have that piece as part of it, we will attack it and knock it down before it gets a chance to get inside and cause trouble. Okay. So that's the whole deal with that. So let's take 10 minutes, get something to eat and drink. And then when we come back, we will finish this part of the viral chapter. Okay. So I'll see you
I
guys.
have one-
Yes, question?
Yes. So my access code expires today, but I don't have one. So what?
Well, what I'm going to take care of that today. I'm going to contact the rep and get those codes for you. Okay.
Okay.
Yeah. And don't worry if there's a delay between submitting the assignment and getting the code. I won't, if there's a problem with the code, I won't hit you with a late penalty or anything. We'll just hold on till the code shows up. Okay.
Okay.
So don't worry
Thank you.
about that. Yeah. All right, Joe. I'll see you
Okay.
at
Thank you.
9:50. Okay, we're back. All right. Okay. Assembly. Pretty straightforward, right? Is putting the thing together, right? Have to do that in order to let it be released and go off and do its work. Release is the number of viruses that are kicked out by an infected cell, and that's going to be proportional to how big the virus is, right? So what we would say is bigger viruses equals fewer viruses. Okay, and that makes sense. It takes longer to build something large than something tiny. How healthy the host cell is, right? So 6 cell, fewer viruses. A pox virus cell, 3 to 4,000 particles. Poliovirus, 100,000. So obviously, poliovirus is smaller. Okay. So we can very quickly generate lots of virus to go out and hit lots of healthy host cells, even for large viruses. So you can see here just a cartoon of what's going on, right? There's your absorption, right? Hookup. We come in through endocytosis in this case. Inside the vesicle, we unpack and unload. And then our genetic material is free. And depending on if it's DNA, it's going to go to the nucleus. If it's RNA, it's going to stay out here. Okay. And then translation happens, right? We make new capsids with genetic material in them, and then we get released. And in this case, we're taking some host membrane with us. So this would be an enveloped virus. Okay. So there it is. Okay. How does this hurt the host cell? Well, virus damage is going to change the way it looks. Okay. So the shape and the size will change. Inside the cell, you're going to behave differently. You're going to be producing virus instead of doing your normal job inside the body. Inclusion bodies are compacted mass of viruses that hurt the host cell. And syncytia are actually fusion of host cells together so that you make this one large multinucleate cell that becomes a virus factory. And then eventually what will happen is that the host cell will die. Okay. So what causes the problem is either the cell can't do its normal job, or cell dies. Okay. And that's what makes the host suffer. In a persistent infection, the cell is going to have the virus, and the virus won't immediately go into production mode. That's lysogeny. Okay. It can remain for the rest of the host's life. It can sit there in the cytoplasm in an inactive state. Okay. lysogeny. Okay. A provirus incorporates into the DNA of the host. Examples include measles virus. A chronic latent state is where periodically the virus gets activated under the influence of certain stimuli, okay? Herpes simplex and herpes zoster, right? This we talked about before. This is the agent of chickenpox. And shingles. Okay, it's herpes zoster. Okay, and simplex is just a family of viruses that sort of use the same method to get inside their host target. Okay. Experts think that about 20% of cancers come from viruses through a process called transformation. All right. So how does this happen? Well, what's going on is that cancer-causing genes called oncogenes okay, cancer genes. They come in with the virus genetic material and they turn the normal cell into a cancer cell, okay? Sometimes the viruses from these oncogenes make proteins that lead to cancer, okay? A transformed cell will have an increased rate of growth. That's a cancer characteristic. Changes in its chromosome. That's another cancer characteristic. Changes in cell surface molecules sometimes. Okay. And can divide indefinitely. That's a cancer characteristic also, okay? So the way that cancer kills you is that the cancer cell grows and grows and grows and travels all over the body, and it steals, so grows and travels and steals nutrients and breaks down healthy tissue. Okay. And that's what kills the host, right? We invade, we chew up the good stuff, we steal food from the good stuff, and pretty soon the good stuff can't work anymore. The initial area of growth is called the primary tumor, right? Primary tumor. That little symbol means primary. Okay. And then it can travel around and land in other places, and those are what are called secondary tumors. Okay. So some viruses that make tumors include papillomavirus, herpes virus, Hep B, HTLV1. There's others, okay? But the reason is simple, right? We get bad instructions in there and we change the cell into a cancer cell, okay? Viruses that hit bacteria include bacteriophage, which that literally means bacteria eating. And most are double-stranded DNA, but there are some RNA phage. Every bacterial species has a phage that will attack it, and they can turn the bacteria from a bacteria that won't hurt us into one that can cause disease. Okay, so that kind of stinks. An example is a T-even bacteria phage. Okay, T-even would be like, they would designate like T2 or T4. Okay, all right. They can hit E. coli, They have an icosahedral capsid. That's this thing here, right? And there's DNA inside it. There's a central tube surrounded by a sheath. Okay. There's a collar. Okay. A base plate. And tail pins. Oops. Those are tail pins. Okay. Tail pins. And then there's fibers. Okay. So see what we're doing? Here we come. Here's our E. coli cell. Bacteria. Okay. So here comes our phage. We hook up. We inject our genetic material just like a syringe, right? So this is absorption. This is penetration. But see, in this case, the capsid doesn't go in. It stays on the outside, right? And then duplication of the genetic material, right? And then eventually we're either going to go to what we call lytic or lysogenic replication, right? So if we're in the lysogenic state, what happens is we just sort of integrate the genetic material into ours and we kind of sleep. But in lysogenic, what we do is we run out tons and tons of viral genetic material, and then we continue down here to build capsid. Capsid builds up, and then we can either be extruded from the cell, meaning leak out, or we can lyse the cell, meaning blow it up. Okay. And often that'll depend on how many host cells are around. Okay. Temperate phages can do either one. They can do either lysogeny or lytic growth. Okay. They do absorption and penetration. They don't do replication or release right away. The viral DNA hits an inactive prophage state, gets into the chromosome, gets copied when the bacteria reproduces. Okay. And so it just kind of lives there, hitches a ride, right? In induction, the lysogenic cell goes into lytic growth. Activated means lytic. Okay. And then what'll happen is that the cell will blow up and the virus particles will be released, or the virus particles will be extruded. Okay. So what happens sometimes is that you can get one of these bacteriophages that bring in a nasty gene to a bacteria that normally doesn't cause trouble, right? These would be things like toxins or surface proteins that can produce ill effects, right? Lysogenic conversion happens when a bacteria gets a new trait from a temperate phage. It's in lysogeny. Okay. So what these are, you can think of them like as guest genes. And they create pathogen. Okay. Examples include Corynebacterium diphtheria. It has diphtheria toxin. Vibrio cholerae, which makes the cholera toxin, and Clostridium botulinum makes the botulism toxin. All those came from phage, right? That hit bacteria that normally don't cause trouble. Okay, we'll talk about how we deal with virus in the laboratory. Viruses have to have a host to do their thing, right? So that's their medium. That's how we make more of them. We can use lab animals or embryonic tissue. That's in vivo culture. In vivo means in life. Okay. Or we can use cell or tissue culture as in vitro methods. In vitro means in glass, like in a glass container. Okay. Why do we do this? To look at the virus in the lab and study it and try and understand how it does its work so that we can design a way to go after it, to help make vaccine, and to essentially understand how the virus does its work so that we can identify places where we can cut into that process and keep it from happening. Okay. So in an in vivo approach, we've got mice and rats and hamsters and guinea pigs that we can use to grow virus. Sometimes we can use insects or non-human primates. Viruses have host specificity, so certain animals can make more virus and others can't, right? So you might think, well, gee, it's mean to use a mouse or a rat or a monkey to do this. Well, that's the only way that we can make more of the virus, right? Sometimes you have to use a live animal because you can't get tissue from that live animal to grow in what's called tissue culture, which would be in a plate or in a container. Okay, so you have to use the live animal in order to make more of what you're studying. Okay, otherwise we can't understand how to fight it. Sometimes you can make virus in tissue culture, right? You can infect not an organism, but you can infect cells from an organism that are growing in a plate or in a bottle that has media in it. Okay. Bird embryos are an example, right? Fertilized egg will have a bird embryo in it. It provides a sterile environment that has its own nourishment. So we can use chicken, duck, and turkey eggs for inoculation. We make a little window in the eggshell, and then we introduce the virus, and it attacks the embryo. Okay. Cell culture is where you take animal cells and grow them in a liquid medium. They attach to a plastic surface. So they're not in an egg or inside an animal. They're in an incubator, in a container. Okay. And they're given everything they need to live. And the cells will form a sheet on the bottom of the tissue culture vial. Okay. It's a kind of an unusual little, sometimes they're round, like in a bacterial plate, little plastic plates, and the monolayer would be on the bottom of the plate, sticking to it, right? Right. But sometimes, like if we look at it from the side, it's like a bottle that's flat, but it has like a crooked neck to it. Okay. Like that. And then the cells grow here on the surface. And then there's liquid media on top of them. Okay. So you change media when you need to, and then you can harvest virus from when the virus gets released into the media, and then you can study it, right? So this is tissue culture. Okay. And so you get your virus from that, okay? This lets us look at the culture and see signs of infections, like for instance, in a tissue culture for a lytic virus, right? What you would see is a sheet of cells. If you look at the bottom of the flash through a microscope, you see a sheet of cells. These cells here, okay? And then you might find some areas where the monolayer stops, right? And there's like a little blown up area around there. And that is what we call a plaque. Okay. Okay. And then you can take virus from the plaque because it'll be in high concentration where this cell just exploded. Okay. You just pipette a little bit of liquid from over the plaque and there's your virus in there. Okay. So you can see some examples, right? There's a plaque. Okay. And there's normal. Okay. This is tissue culture. Okay. So we would call that a CPE, right? Microscopic manifestation of a cytopathic effect. CPE is the plaque, right? The area that's destroyed on the monolayer of cells. And you might think, well, why can't we just do this for everything? And we don't have to use an embryo or a mouse to do any of the work. And the answer is that not every virus for every host has cells in it that can be grown in a tissue culture. Okay. They just don't grow. And we're trying ways to make them grow, but we really haven't figured out for some of these how to make that happen yet. Okay. You have to have a recipe for tissue culture that lets those cells grow outside the animal and keeps the cells healthy and also keeps the cells acting like cells from the animal. And that usually means you have to have in the growth media nutrients and electrolytes and all that, but you also have to have things called growth factors that make the cells behave like they do in the animal. Okay, and those are little proteins that that can come from the organism or can be made by copying the information that comes from the organism. You can buy them in a catalog. And for some cells and tissues, we have that. For some, we don't. The same technique is used for bacteriophage, right? What you do is you get a plate, okay? You get a plate here, and then you, just like we saw in the aseptic video, right? We inoculate. Okay. So we spread liquid over the surface. This is media. Media. Okay. We spread liquid over the surface of the media, all over, right? And then we grow a sheet of bacteria, right? Color here. I don't know, gray. Right, so now we have bacterial growth, a sheet all over. The whole thing is just filled. Okay. So you don't have colonies in this case. What you have is just one big lawn bacteria, right? And then you add your phage, right? Usually is a liquid from something somebody gave you, phage, you're floating around in it, had the phage. And then you wait, and what you end up with are, after the phage does its work, you've got areas in the lawn that are clear. Those are the plaques, okay? And every other area is still covered in lawn. Right? So just imagine these holes there. This is all lawn. Okay. And where would our phage be? Our phage would be in the plaques. So you can just pick the plaque and use that to do more experiments, right? Because your phage is going to be in there. Plaques. Okay. And those are just areas where the host cells have either been slowed in their growth or in some cases where they've burst open. Okay. There's all kinds of virus in that area. Okay. Okay. Other agents besides viruses that can cause disease. Okay. This is one that has some important ramifications locally. Okay. Spongiform encephalopathy is a fancy term for a disease that turns your brain into jelly and destroys it. Okay. There's usually a latent period before the first signs appear, and the disease is fatal. It'll kill you. Okay. This is mad cow. All right. Caused by a protein that you can ingest, you can eat in infected meat or bone or nerve tissue that is so tiny that it can pass through the lining of the digestive system and get into the blood, and from there get into the brain. And what it does once it's in there is it makes other proteins misbehave by folding them up wrong. And when it does that, the cell's no longer functional and it dies. Okay. And so this is what destroys the nerve tissue from the inside out. Remember that nerve tissue doesn't regenerate. So once it's gone, the party's over. Okay. The human disease is called Creutzfeldt-Jakob. Okay. This is what's in humans. You might say, okay, well, how did we know this? How did this happen? Well, it's kind of an interesting story. Okay. This disease, this Creutzfeldt-Jacobs disease, was first described in New Guinea. Okay. And what would happen is that typically the mothers and the children would come down with this debilitating condition that would kill them in about 3 days. It would just melt their brains and they'd be dead. And they thought initially that maybe it was genetic. Okay. And so what they did was they collected information from the tribesmen and tried to determine, who had the gene for this disease and passed it down to the offspring, but nothing that they came up with made any sense. And then what they found out later on, after they collected enough information, is that the disease tended to show up in families where the head of the household had recently died. So that was a clue, right? Head of the household dies and suddenly this disease pops up. So what's going on? Well, it turns out that the New Guinea tribesmen used to honor their dead by eating them. They were cannibals. So when the head of the household would die, they would prepare the body and eat the whole thing head to toe. Okay. And that was supposed to honor the head of the household. And what happened by doing that is that they ingested nerve tissue, which is where the prion lives. Okay. And then the prion would soak in to their blood through the digestive system and spread from there to their brain and would melt their central nervous system in a few days and they'd be dead. Okay. Well, around here, with mad cow, that's the bovine, that's the cow version of the disease. And we've traced the cause of that to putting bone meal in the feed. Okay. So bone meal plus cow feed can lead to mad cow. So what's going on there? Well, turns out inside the bone meal is nerve tissue. And that's where the prion lives, okay? So the cow eats the bone meal has the nerve tissue has the prion in it. The prion gets into the cow and it kills the cow, but it also infects the meat. And so if you eat the meat from a cow with mad cow and it's not well done meat, you can get the human version of the disease and die from it, okay? All cases of mad cow have been traced to meat that's come into the United States from outside the US. Those are cases here in the US. And that's the reason for it, is this farming practice. The reason that they put bone meal in the cow feed is to increase the protein content and make more meat faster. But unfortunately, you can end up with this problem, okay? We don't do that in the United States. We only feed our cows grass or grain, right? That's what it says on every steak from Kroger's, 100% USDA grain-fed beef. Okay, which means it's never augmented with stuff like bone meal, okay? It's all plant-based. We have a problem in the tri-state called mad deer. Okay, same deal. Okay, it's mad cow disease in the deer. Now, how's this happening? Well, the deer overpopulate. And the reason for that is we don't allow hunting. Okay, the deer overpopulate and they go out into urban and suburban areas. I live in Clifton. I can see deer at twilight walking down the street and in my backyard. They're all over the place. The reason you can't normally see them during the day is that they blend into the background because they're the exact color of mud, right? So they're almost invisible to your eye. And when they leak into these urban and suburban areas, what do they eat? Well, they eat garbage. And they also take in dirt that's had garbage lying on it. And in the garbage and in the dirt can be nerve tissue from animals that have previously died, right? And have left their remains there, right? And the result is they can pick up the virus and they can start acting funny, right? You'll see stories every now and again of a deer jumping off a highway overpass into an oncoming vehicle or walking through a plate glass window in the middle of downtown, okay? This is not something that deer normally do, okay? And what it indicates is that their nervous system is breaking down. And if you eat venison from a deer that's affected with this, you can pick up mad deer, okay? So this is something that's even smaller than a virus, right? So this is maybe a nanometer or two wide. And that's why it can get through so many barriers in the body, right? Because it's just so darn tiny. And it can survive the acid of the stomach and slide right through and get into your system. Satellite viruses are just viruses that need normal virus in order to make more copies of themselves. So they're sort of like incomplete viruses. Okay. Examples include adeno-associated virus and delta agent, which is only expressed when hepatitis B is around, and it makes the liver damage much worse. Okay. So these are literally just either genetic material or hunks of DNA that can be reproduced when there's normal virus present. Viroids attack plants. They're a tenth the size of a virus, and they're just RNA without a capsid or other type of coating. Significant pathogens include viroids that attack tomatoes, plants, citrus trees, cucumbers. Now you might wonder, well, how in the heck is just a piece of genetic material getting inside a cell? Well, it turns out that even a molecule, a piece of genetic material, can sneak into a cell if the cell membrane has pores in it, or if it just gobbles up the material through endocytosis. Okay, this is one of the ways, for instance, that you can transform a bacterial cell with a phage or a bacterial cell with a plasmid, is by turning it into a, almost a salt, a mineral, and you throw it at the bacterial cell, and the bacterial cell eats it. And then once it eats it, it's inside, and it basically unravels. And now you have genetic material inside of that bacterial cell, and now that's a transformed cell. Okay, so this can happen in nature too. It just gets eaten and it comes in. Okay, so how about designing drugs to fight all this stuff, right? Viruses cause things like cold, hepatitis, chickenpox, flu. Prominent viral infections include dengue fever, yellow fever, and of course, COVID, all right? AIDS and Ebola are types of viral infections that have high mortality rates, meaning you get these, you die, okay? And long-term disability can come from polio or rubella. They can have a connection to chronic infections like type 1 diabetes or cancer, or even Alzheimer's, we think, okay? How do we treat? Antibiotics treat bacterial infections, and they don't have an effect on a virus. Antiviral drugs are going to go after viral reproduction, but they're going to have side effects because that's going to hit our normal tissue too, right? So that's collateral damage. And we know the reason for that, right? Because the host does a lot of the work here. Example, double-stranded RNA caspase oligomerizer is an example of an antiviral. Virus-infected cells destroy themselves no matter where the virus is, okay? And so what this does is it just chops up the instructions that are responsible for making the capsid proteins so that they're useless, okay? It's a little bit like what interferon does for us. When interferon goes into a normal cell, the cell makes a protein called dicer. And what dicer does is it chops up double-stranded DNA wherever it finds it. And that's going to interfere with these RNA viruses' ability to do their work. There are vaccines, but again, not to everything. Now, why is that? Why don't we have a vaccine for every viral disease? Okay. The answer is very simple: mutation. The normal idea behind a vaccine, right, is a vaccine has a piece of the pathogen or killed pathogen okay, you inject, okay, and that makes you, it causes you to make white blood cells that attack the virus. Sounds good, right? But it fails in a lot of cases because viruses can change their appearance through mutation. Mutation causes changes, the triangle means changes, in surfaces of the capsid surface. And then immune system has to start over to attack the mutant. It's like coming in with a disguise, right? You come in with a disguise from a mutation, your vaccination won't be effective because it's to a different type of molecule than the one that's being carried on this new invader. And the reason that viruses can mutate so much is because they don't require all the other things that most pathogens need to reproduce. Yes, Benita.
So isn't the COVID virus mutating, or
It is. There are mutants out
you
there.
think
That's
they
why
made
you
these
see.
vaccines a little
That's why
too
you see
early?
articles. That's why you see articles about the concern about what they call variants, right?
Mhm.
Variants or mutants. So what they're doing right now is they're taking these variants and they're seeing if the current vaccines are effective. And of course, that takes time. Okay. And we won't know whether or not it's effective against variant until the information from those studies come in. So that's one of the reasons why, for instance, when you get a flu shot, you can get a flu shot and then you might go out there and you might get the flu. And you'd say, well, wait a minute, I got a flu shot. Well, the flu shot is just the best guess of what's going to be out there that year. And it's not going to be able to know whether or not there's going to be a mutant or a variant, right? Because you can't know that. You can't know that till it happens. So you just make your best guess, you take your shot, and then you're going to be healthy against most of what they think is going to be out there. But there's going to be people that are going to get the flu because they're going to run into variants, okay? And viruses do this much more easily than living things because they don't rely on tons and tons of information to do their work. They just need a few genes in some cases. And then the other stuff, the protein genes, there are changes that can happen quickly that can still result in a viable capsid, an infectious particle, right? Just it looks different. It's like painting it a different color or putting a different shaped spike on it or something. But it still can infect, it can still produce disease. And so your immune system has to start from zero. So, but that requires that the immune system find the white cells that can attack and knock it out, then tell those cells to make an army, and then that army goes to work and does its job, right? And then you're resistant from that particular antigen from that point on. But that doesn't mean another antigen couldn't show up, right? So it's always a
Right.
race between your immune system and what's going on with the pathogen at the same time. It's like an arms race, right? You do this and I'll do that. You do this and I'll do that, okay? So in the long run, it's good to get the vaccines,
Mhm.
but no vaccine is going to be foolproof against a virus. There are vaccines against bacteria that cause disease that are very good because bacteria don't mutate nearly as quickly as a virus can. And that's because bacteria have to do the whole process of reproduction and growth using their own information, right? Because they don't take over a host in the sense that the host makes more of the bacteria. They attack the host and break it down to get nutrients, but they're not really in there to use the host to reproduce, okay? So that's the game, right? And that's why, so it's not a conspiracy or anything. It's just that viruses they're tough. They're tough. They're like a vehicle that causes disease that we have yet really to figure out a way to conquer effectively, right? There's all sorts of approaches that you can take if you can identify vulnerable parts of the virus. Like you might be able to make a drug that goes after how the capsid pieces fit together, right? You can maybe bind to that and keep the capsid from assembling, or you might go after an enzyme that only the virus uses and makes. Okay, we've done that with AIDS already. That's why we have an AIDS cocktail that's pretty effective now. Or you might go after the life cycle, right? You might go after if it does double-stranded RNA at some point, right? You can get in there and try and knock that out. Okay, so it's tough, right? It's difficult to do a vaccine and get it up and running quick and have it effective every time. Okay, we seem to have gotten lucky with COVID. The nice thing about COVID is it's generally not lethal. 99.9% of people who get COVID survive. Okay, and about 99% of them don't even have major ill effects. Most of the problems with COVID are with people that have what are called comorbid conditions, meaning there's something else already wrong with them that makes them weaker, that makes it easier for the COVID to cause problems. Okay, sometimes it's AIDS, sometimes it's things like diabetes. Okay, there's other conditions that can feed into that. But young, relatively healthy individuals have virtually no risk. Okay, when you look at the number of deaths per the whole population, it's very, very, very tiny. Okay, so eventually what happens with viral infections is that enough of the population gets the infection and recovers that they cloak people that don't have or never had the infection, and that's called herd immunity. Okay, meaning, you know, if I'm living in the middle of a crowd that's resistant to virus X, the likelihood that virus X will be able to get to me is almost zero, right? Because I'm surrounded by an environment that's just going to not let the virus reproduce, right? It's when it's something new that the immune system's never seen before that it can spread quick, like the COVID did. Okay, and then a lot of people can get infected, right? But one of the things that's going on here that you have to be careful about is that they're reporting cases. Okay, cases does not equal deaths. Okay, that's something you have to be aware of, right? That those two things are totally different, right? You can have a case of COVID and have no symptoms at all, right? It's the deaths that matter, right? And then you have to, in the death information, you have to be able to separate death from COVID versus death with COVID. Okay, one of the things that's going on right now is that they're adding these numbers together and giving you the death number, right? Well,
Yeah, and that's stressful.
yeah. Well, so I mean, like you could go in. There's been reported cases that you could go in with a gunshot wound, okay, and die of the gunshot wound. But if you happen to be also infected with COVID, they'll say that's a death with COVID. All right. Well, he died from the gunshot wound. He didn't die from the COVID, but you added those numbers together and made the number bigger than it really is. Okay. And the reason for that is that they're making insurance pay for anything right now that's COVID related. So everybody's trying to make stuff associated with COVID. And so the death numbers are much larger really than they should be. So that's a few of the important things to realize about what's happening. Okay. But the big thing is that for healthy young individuals, it's not a big risk of death at all. Okay. Now, could something come along that could be a big risk of death? Sure, absolutely, there could be. Okay. The Ebola outbreak was much more concerning than the COVID outbreak because the mortality rate for COVID is much higher. Okay. I mean, the mortality rate for Ebola is much higher. You get Ebola, you die, right? The Ebola just melts you into a puddle of goo. And because of that, oddly enough, it doesn't spread very effectively. And you might say, well, why is that? Well, because for a virus to really spread, it needs to hang out inside the host for a while to let the host interact with the population. And if it doesn't do that, if it just melts you, right, then you can't go out and interact with anybody because you died too quickly. The host didn't get a chance to spread the disease very effectively. So that's a saving grace with Ebola, is that it's so aggressive and so rapid that the spread tends to be minimal. Okay. So those are just some of the things to think about when you're thinking about viruses, right? They've been here a long time, and they're tough to deal with. They're very, very challenging to design drugs for after you've been infected, right? But definitely get your vaccinations, okay? And another thing about vaccinations while we're on the topic, and this is important, is that you'll hear stories of people that get vaccinations, and after they get the vaccinations, they get real sick, okay? And that's not that's not because of the vaccine. That's not because they've gotten the disease from the vaccine, okay? It's because they're having what's called an allergic reaction, an allergic response. Okay. So hold on a second. So in every population, you're going to have some individuals that have an allergic response to something. Okay. I could take a population of, say, 1 million people and inject you with isotonic saline, like they use in the hospital. Okay. And you would have some individuals that would have an allergic response to that. Why? Because there's something in the saline that they're, hold on.
Renita, we're still on, right? Renita,
Yes.
okay, sorry. I was just wondering because sometime my computer act up.
Oh, no, we're
It's-
still here.
It's
Just
been
in-
quiet for a long time. I'm like, what happened? I know he said he will come back, but I'm like, am I still on? Okay, thanks.
Yes.
Sorry about that, guys. My wife's in the hospital. Okay, so in any population, when you inject anything, could be a vaccine, could be saline, could be whatever, you're going to have a percentage of the population that's going to be allergic to it because they're going to have white blood cells that see that as an invader and attack it as if it was going to cause harm, even though it's not intended to. And they release something called histamine. And that produces inflammation. And causes tissue damage. Okay. So these people are, they are sick, but they're sick because of their own immune system. All right. Now, the way allergies work is that most people go through life never finding their allergen. The allergen is just the thing that causes the allergic reaction. Okay. And an allergen could be anything. It could be pollen, it could be dust, it could be animal hair, or it could be something serious like a flu virus. Okay. Most people never run into their allergen, and they go through life with an immune system that defends them against disease relatively effectively. But some people, unfortunately, run into their allergen every day. And as a result, they need to take medications that suppress inflammation. Okay. And you've seen them. They're over the counter things that are available for people that want to knock down allergic responses. The flip side of that is that if you're taking medications that knock down inflammation, then if you do get an infection, it's going to be tougher for you to kick it because the inflammation is part of the set of weapons that we use to eventually knock the infection down. Okay, so there's the problem, right? Is that if your immune system is the bad actor, we have to knock it down, and that makes it easier for you to get an infection. And that's one of the reasons when we talk about, once we get to the point where we talk about what are called autoimmune diseases, where your own tissue is the problem, they're very difficult to treat because we have to knock down the whole immune system in order for you to not suffer the ill effects, because we don't have the ability right now, to only take out the white blood cells, those particular white blood cells that are causing the problem. Okay. So when we do that, it makes it more likely you're going to have an infection. So some examples of autoimmune diseases are type 1 diabetes and Graves' disease and lupus. Okay, and we'll talk about all those a little bit later on in the course. Okay, but I just wanted you to be aware of, you know, what's going on when you hear about people saying, well, they gave me the vaccine and then I got the disease. No, you didn't get the disease. You got an allergic reaction. Okay. And that can happen in any large population. They usually list those as what are called adverse events. And that's what you see on the advertisements for a lot of drugs on TV, are the adverse events that have happened from using that drug. It doesn't mean you use that drug, that's going to happen. It means that it could happen. Okay. And that's the problem with any drug, right? It takes a while to make sure the drug is totally good to go. And that's one of the reasons that drugs are so expensive, because you have to do these tests and you have to take the data in, you have to analyze the data, and then you have to figure out, is this a good drug, or is this drug that's going to cause more problems it's going to help? And that ends up adding to the cost of the drug, right? You can't just whip up a drug and then throw it out there and say, take this, it'll work. You have to check it, right? Check it to make sure that in your target population, it's going to do what you hope it does. You know, and if it doesn't, you're not going to develop that drug. You're going to pull it, you're going to try something else. Okay. So I think this is good. Let's stop here. Let's take, we got to 11:30, but let's just stop here for now. And then we'll pick up the other chapter in lab. Okay. So I'll join you guys at 1, and we'll talk about the metabolism chapter at that point. Do we have any questions before we go? We good? I will see you guys at 1 o'clock then. Okay. Come again? I heard something. Okay, I'll see you guys at 1.
Thank you.